Hello, everyone. Welcome to the TIDES Digital Week, brought to you by the organizers of the TIDES conferences. My name is Mike Moriarty. I'll be your host for today's session titled From 10 Grams to Several Hundred, a Case Study in Peptide GMP Process Development. First, I'll cover some quick housekeeping items. If you experience difficulties with audio or advancing slides, refresh your screen with F5. If you are experiencing other issues, hit the question mark button to receive assistance. At any time during the presentation, submit your questions into the Q&A window on the left-hand side of your screen. In 24 hours, you'll receive a link to watch the recording of the session. Let's now begin by introducing our speaker from PolyPeptide, Niall McLoughlin, Development Project Chemist from PolyPeptide. Thank you for joining us today, Niall. Now I'll hand it over to you to begin our presentation.
Thank you, Mike, and thank you to everyone who's joined us here today. I'd like to first begin by thanking the organizers of the TIDES Digital Week for giving us the opportunity to present here from PolyPeptide. Having already watched some of the other talks already, I've been really impressed by the level of science that's been presented, and I think the TIDES organizers should be very happy with what they've organized this week. And I'd also like to thank them for giving me the opportunity to present here today, this morning or this afternoon, wherever you're listening from. So like Mike said, my name is Niall. I'm here on behalf of the PolyPeptide Group, where I work as a process development chemist. So today, the purpose of my talk is really to give you an insight into industrial peptide production and how we develop large-scale processes, going from small amounts of material to several hundred grams, as alluded to in the title of the slide. Before I get into the science, I'd just like to quickly say a little word about who PolyPeptide are. So PolyPeptide are a contract development and manufacturing organization, which are specialized in the production of peptides. So PolyPeptide produce around half of all of the approved peptide APIs currently on the market. We're also involved in the production of generics and also oligonucleotides. And I've showed a few of the peptides that we produce in generics, also on the right-hand side of the slide here. So given our position, we work quite closely with large pharmaceutical companies, but we also have very strong ties to the biotech sector. We work with startups, and we also work with academics in order to produce peptides at various different scales. So we work across a number of different sites around the world to produce peptides. So we have 3 sites in Europe, we have 2 in the US, and we also have a site in India. So I'm coming from the PolyPeptide Strasbourg site, which is a bit of a special polypeptide site in terms of the network. That's because we're what's called a pipeline builder site. So that's to say that we typically get in projects that are like quite early stages. Maybe there hasn't really been an industrial scale synthesis or production before. So we'll take in a project and we'll develop the peptide synthesis, the production, the process route from a very early stage. So we'll generally develop the first, do the first process development. Then we'll go through maybe several rounds of non-GMP synthesis or GMP production in order to get to a process that we're really happy with, and that's quite optimized. And from there, then we generally transfer the process then to a larger site in the network that can produce it on a very large scale. So we're really where a lot of the projects begin in terms of the polypeptide site. So in Strasbourg, we're FDA inspected, we're a GMP site. We have several different synthesis and purification lines, so we can handle multiple projects at the same time. And generally, we say that we can produce anywhere from several milligrams to several kilos of a given peptide. So aside from development and production activities, we are also quite lucky to have the Center of Innovation for the PolyPeptide Group in Strasbourg. So they're involved in many various activities, such as green chemistry, and also looking at how new process technologies can be implemented in producing peptides. And we're also quite lucky to have the global sourcing department of the PolyPeptide Group here. So they're a special team of synthetic chemists who are specialized in producing strange and exotic amino acids and building blocks and things that might be quite difficult to source elsewhere for peptide production. So that's who we are. And just going on to the actual topic of today's talk. So I wanted to give you a bit of an introduction into how we go about developing a process. So I've taken an example from a project that I worked on over the last year or so. And here it's a case study where we had a new client who came to us and they wanted to have 100 grams of peptide manufactured to GMP quality standards. So when we get this kind of request, this kind of order, then where do we begin? The first part, probably most important, is that we try and gather as much input from the client as possible. We try and to assess their need in the best possible way. So firstly, we ask what is the application of the product itself, and when is it required. So in the case of this client, the peptide was required for phase 1 clinical trials, and it needed to be delivered by the second quarter of 2022. We also then ask what kind of criteria need to be met in order to actually perform this application, so the phase 1 clinical trials. So in this case, the client needed the peptide to be greater than 95% pure according to a validated analytical method. It needed to be an acetate salt, and we needed to verify its identity by MS and also amino acid analysis. And we also needed to, at the end of the campaign, quantify and identify any residual materials or impurities in the product. This can also include things like water, TFA, trace organic solvents, and also we have to prove that the final peptide product is free of bacterial endotoxins, yeast, and mold. So once we've gotten this information and we've gathered the essentials of the client's requests and needs, we then actually look at the peptide itself and we assess what might actually need to go into the process development and what we need to take into consideration in order to produce the peptide. So here, the chemist, so this is generally my job, we sit down and we actually look at the peptide sequence itself and we assess what needs to go into its production. So we'll consider things like the solubility and stability of the peptide, if there's any sensitive or aggregation-prone sequences in the peptide itself. Is there any need to include special building blocks, synthetic elements, special linkers? Is there any coupling strategies or deprotection strategies we should consider? And then we'll also consider things for downstream processes. So what kind of cleavage conditions we might use, what kind of stationary phases are required, or what kind of ion exchange strategy we might use in order to get to the target salt. So in the case of this peptide, for this client, the peptide was relatively simple, I would say. So we had a linear peptide of 25 amino acids. It only contained canonical amino acids, and it had standard N and C termini. So there wasn't anything too exotic in the structure and the sequence. That's, for me as a chemist, pointed out a difficult or maybe quite involved process development. So just as a backup to this, what we enact in PolyPeptide as well is that we have the chemist assess the peptide, and that's based on internal knowledge, and it's the expertise of the chemist that comes into play. But we also have informatic tools which have been developed internally, which also assesses the peptide to see if there's any kind of process development considerations that also need to be taken into consideration. So this is an informatic tool which has been developed internally, and we've published about this tool as well. But essentially this takes into consideration the physiochemical aspects of the peptide. It will go through literature to see if the peptide has been produced in other ways before, if there is anything maybe critical about the sequence. And it will also take into consideration peptides that have been produced already internally according to an internal database. So basically this tool is implemented to basically complement the expertise of the chemist, and like that we can really minimize any kind of process development iterations or catch any things that might be unexpected in process development. And this overall helps us to secure the purity and also maximize the yield of the process before we even start the real development. So once we perform this internal knowledge assessment, we then go to the client and we propose a development and manufacturing plan. So in the case of this peptide, we propose to perform what's called a demonstration batch of about 10 grams of the peptide. So here we propose to perform a rather generic synthesis, so use very classic amino acid coupling conditions to reduce the peptides. From there, we would generate a crude, which we would then use to perform development of the cleavage from the resin, so that we use during a solid phase peptide synthesis. And from there, we would then define a purification strategy based on the crude, which we obtained from the cleavage development. So really the purpose of this demonstration batch is to really test and optimize representative conditions for GMP production. It also helps us then to establish yields for each process step, so that's really important in understanding how much we actually need to put into our process, so the GMP scale that needs to be considered. And then, of course, by performing this test scale production, it allows us to also identify any challenging aspects of the process that might need to be taken into consideration before we move forward with GMP. So the global idea, the core points of performing a demonstration batch is to really assure that we produce the peptides to the quality and the quantity that the client has desired, and also to produce it in time so that we avoid any errors along the way. So that's basically what we proposed to the client. We started our demonstration batch of 10 grams by first performing upstream development. So this is the actual synthesis of the peptide. So here we use very classic solid phase peptide synthesis conditions. So we performed the synthesis on a 25 millimolar scale using a preloaded Wang resin with the first amino acid attached. We then performed amino acid couplings using very standard dicoxyma conditions. We monitored every coupling until completion using a colorimetric testing. And then we washed the resin after coupling in batches with DMF. After the coupling of each amino acid, we then deprotected with the piperidine DMF mixture. And then after deprotection, we washed the resin using a method called percolation, which has been developed internally in PolyPeptide. So I'll explain what that is in just a second. But just to really enforce the idea behind this demonstration batch, what we're actually trying to do by producing this peptide on a smaller scale. So we're looking at things like coupling durations. We want to measure things like the resin height to see if the resin is growing or shrinking during the synthesis. We are trying to measure how much solvent we're using as well, because this will be important when we scale up for GMP. And we're also trying to characterize any impurities that can be formed along the way. So we're really trying to identify if there's any tricky aspects to the synthesis that we need to watch out for before we scale up. So just to go back to this percolation thing that I mentioned briefly a minute ago. So typically after a deprotection of an amino acid, after a deprotection of a peptide on the resin, we wash the resin to remove any residual piperidine. So the typical way that you might remove the piperidine is by performing what's called batch washing. So in this case, you have your resin, your deprotection solution, that's mixing during deprotection. You drain it at the end of this step. You have your drained resin, and then you add a wash volume of your DMF. You mix it with the resin again, and then you drain it in order to remove more of the piperidine. Generally, you have to perform this several times in order to actually get down to levels of piperidine which are below specification. So we monitor how much piperidine is present in the resin at the end of each deprotection step. So generally, we found that this will take probably about 5 or 6 washes in order to get to a quantity of piperidine that's actually satisfactory. So what we've done in PolyPeptide is we've studied this in quite a lot of detail, because as you can appreciate, performing such washes, 5 or 6 washes after each deprotection, it costs a lot of solvents, and also that has quite a large environmental impact. So our idea was to improve this, and to do so, the idea was to perform what's called percolation. So we have our deprotection as standard. Resin and deprotection solution are together. We drain it. But then at the end of it, we leave a partial volume at the top of the resin. So we leave the resin swollen, and then we add volumes of wash solvent in a continuous flow, continuously draining from the resin, in order to very smoothly in flow remove the piperidine altogether at once. So you can see here, by adding the volumes of wash solvent in a continuous flow and gently draining, we much more efficiently remove the residual piperidine that's attached to the resin solution, the resin itself. So the idea here is really a flow-based method rather than a batch-based method. And by performing this percolation instead of a batch wash, we find that we can generally reduce the amount of DMF that we require from anywhere to 20 to 60% of volume itself. So this is quite significant when you consider that in some GMP campaigns, you're really using thousands of liters of DMF for an overall campaign. This can be quite significant in terms of savings and in terms of the economy on the environment as well. So that was just a brief aside about what percolation is. So to go back to our actual upstream development and our demonstration batch. So we got to the end of our synthesis. So it was a 25 amino acid peptide. We had a HPLC purity according to our generic methods of about 73%. And we noted that the peptide was synthesized relatively well, but we had 1 or 2 impurities mainly associated with what we call endo impurities. So these are the double incorporation of amino acids at the same position as you can complete the synthesis of the peptide. So here we had a number of ones such as endo-ala, endo-valine, endo-gli as well. And we associated these endos, these double incorporations, with overnight couplings, so couplings that lasted significant amounts of time. And we also saw in certain cases that we had endo incorporations where the resin wasn't coupled overnight, but it was stored overnight in DMF after being washed twice. So we noted that these impurities would probably have to be dealt with before we move on to the GMP campaign. So these were impurities that we thought we could deal with in a better way. So in any case, we took the crude resin that we produced from this demonstration batch, and then we performed a screen of different cleavage cocktails in order to move forward for the next step. So we tried different cocktails, and we saw that they had relatively similar results. So we took the cocktail that gave us the best HPLC purity upon cleavage. We performed this first test at 100 milligrams of peptide resin, and then we moved up and we performed tests at 5 grams and then also at 75 grams. So as you can see here on the right-hand side, in terms of HPLC purity, we have largely similar results from our initial 100 milligram scale in terms of purity and also in terms of yield. So you can see that for this cleavage step, we have quite a robust and scalable process step. And for our first batch, we are quite happy with this part. So taking the material that we cleaved from our 75 gram cleavage, we then looked at the the crude purity of the peptide according to the client's analytical method. So here, by looking at the HPLC chromatogram, we see that a lot of these endo impurities, so the endo-allo, endo-ala, the endo-gli, and the endoproline, they came very close to the actual peptide peak itself. So we saw these as critical impurities that we'd have to really work on in terms of purification downstream. So these were really the impurities that we sought to get rid of. What we envisaged for this purification was to perform a two-step purification strategy. So the first step would be, of course, performing a primary purification by reverse phase HPLC to essentially get up to 95% purity. And then this would be performed in a TFA-based system. So, and then after this, we perform an ion exchange in order to exchange the salts for an acetate salt, which was desired by the client. Then performing lyophilization in order to get the final peptide powder at the end. So this is what we envisage for the actual purification. But in order to define conditions, we generally perform what's called a purification screen. So to do this, we generally start with what we consider our generic conditions for a peptide purification. These conditions are based on internal knowledge, but they're also based on preliminary experiments that we perform in the lab, where we assess the crude and where it elutes, and also certain other conditions. And then we perform very small-scale purifications, generally with 10 or 20 milligrams of crude peptide per test. So from here, each parameter which you see in this box, which discusses the generic purification conditions, we modify each parameter, testing various purifications in sequence at this 10 or 20 milligram scale. And from here, we assess the quality of each purification in terms of how pure the pool is at the end, what kind of recovery we have, and what kind of preparative profile that we have for the purification. So in the case of the mobile phase, we screened 5 different conditions, and we saw that at 0.05% TFA, we were able to get the best of these conditions, the best of these results from this condition. So moving on from there, we then screened the other parameters. So we screened for the stationary phase, our loading, and also the gradient. And by virtue of this process, we're able to get to HPLC purity of the best fraction at 96%, and also a pure pool purity at 95.3%. So we're able to attain the specification that we were looking for the primary purification. After this, we then performed a very similar strategy for the ion exchange. So again, we screened different stationary phases, and we also looked at different mobile phases. And so at the end, we're able to get a pure pool purity of 95.6% as well from this. So again, this is all being performed at quite a small scale, about 10 or 20 milligram scale. But then, of course, we need to also assess how we scale up. So for this, we took the crude, which we had cleaved previously from our 75 gram test, and we purified the 36.7 grams of crude that we had from this test according to the conditions which we defined in the previous slide. So we performed first a primary purification, where you can see the chromatogram on the left-hand side. And after that, we implemented the ion exchange conditions and lyophilized the peptide as previously described. So in the end, we were able to get a final HPLC purity of the peptide at 95.3%. So this is within the specifications desired by the client. We were able to obtain about 11 grams of the peptide as a dry powder. And we were also able to meet the other analytical specifications that the client requested as well. So the peptide was isolated as an acetate salt. We were able to confirm its identity by mass, by elemental analysis and amino acid analysis. And we also found that in terms of residual solvents, that we were all within specification. We had very little remaining at the end. So from the overall process, if we take all the steps into consideration, we had an overall yield of about 25%, which is quite good and quite robust in terms of a demonstration batch, in terms of a first try essentially. But we did note that for their primary purification, our yield was relatively low, about 31%. And this mainly comes down to the fact that we had quite a lot of these endo impurities present in our crude. So this made the primary purification step a bit difficult. We did have to sacrifice some fractions that contained pure product. So for us, in terms of the development, so in terms of the demonstration batch, we had a process that worked, that was robust and scalable, and that we were happy with. But there was one or two adjustments we would have to make in order to go to the GMP batch scale. So moving forward with this, moving from the demonstration batch to the GMP batch, we have to perform a few different optimizations and also calculations. So in one sense, we have to calculate the scale of the GMP batch that we want to use. So we take into consideration all the yields of each step that we previously tested during the demonstration batch. We take into consideration the amount needed for the client itself. And we also take into consideration how much is required for QC testing, for regulatory reserve and other tests. And generally, we take into consideration a worst case scenario. So we have an overall yield that doesn't include things like fraction recycling, which might be able to save us in certain cases. So taking all these different factors into account, we estimated that we would require 325 millimoles in order to produce the 100 grams of peptide required by the client. So based on this estimation, then we performed our raw material sourcing. And of course, in terms of GMP campaign, this equates to a lot of DMF. So we're over 1,000 liters of DMF, we have 50 liters of piperidine, and almost a kilo of silica. So you can really appreciate the kind of the increase in scale that we're really doing when we're going from this small scale demonstration batch to a GMP campaign. After then, we also took into consideration what we noted during the demonstration batch, little things that we might want to improve. So the big improvement we wanted to address, or the big point that we wanted to address, were these endo impurities that we noticed. So to address these, we considered increasing the number of batch washes after amino acid couplings. So this was to address these endo impurities we saw when the resin was stored overnight. And in the case of glycine, proline, and alanine, we decided that we would limit overnight coupling. So we would have a fixed time for each coupling, and we wouldn't let them run overnight in order to avoid also double incorporation. What we also did for the GMP campaign, and that was also requested with the client, was introduce a UPLC method to assess fractions and final impurity. So this validated analytical method was developed in between the demonstration and GMP batches in order to provide a better assessment of the purity during our GMP campaign. So taking all these different considerations into account, we then actually put together our GMP documentation. So this, of course, is a very important step in any GMP campaign. So we put together our master batch record. This is a detailed step-by-step protocol of each step in our production, so our synthesis, our cleavage, and purification. In the case of this campaign, we came over 400 pages for the entire document. And of course, this document has to be read by our production team and also our quality team to ensure that it's honest and it's correct. And it's also read by me in terms of ensuring that technical aspects are respected. So once we had everything in place, we had our raw materials sourced and our GMP documentation together, we then moved forward with our GMP campaign. So as before, we performed our synthesis on a preloaded Wang resin. Of course, this time we're now at 325 millimolar scale. So we're moving up from a 1 liter reactor to a 10 liter reactor. And just to cut a long story short in terms of the synthesis, at the end of our synthesis, we produced the 25 amino acid peptide, and we saw that we had a significant reduction in the percentage of endo impurities, having enacted the various steps which I described in the last slide. So this increased washes and also reducing the coupling times of certain amino acids. So in one case, we were able to eliminate the presence of the endo-alanine impurity, which we saw previously, and also other impurities were greatly reduced. So if we compare the crude purity in the chromatograms at the bottom of the page, we can see that compared to our demonstration batch, we go from a 4.1% presence of endoglycine to a 2.5%, and also we see other impurities like endoarginine and also endovaline have been reduced as well. So we have a crude purity now of 82.9% versus a crude purity previously of 67.1%. So we were very happy with this. And we thought that this would greatly help us in terms of our purification downstream. But first, of course, we have to cleave the peptide from the resin. So as compared to our demonstration batch, now we're cleaving over a kilo of peptide resin versus 75 grams. And this allowed us to obtain about 700 grams of crude peptide from the resin. Again, our crude purity was improved compared to the demonstration batch. And we had a very similar yield. So we see that this process is very robust and very scalable, given the changes in the amount of peptide resin that we're implementing here. So moving forward, we then performed our primary purification. So again, this is implementing the same purification conditions that we defined for our demonstration batch. And you can see here that in terms of purity, we're also improved compared to the demonstration batch. So we have a 96.9% purity versus 95.3% for our demonstration batch. But the real difference, and this was even surprising to us, is the recovery yield that we're able to get from this step. So if you compare it with our demonstration batch, we go from 24.9% to 71.9%. And this is basically due to the fact that we have much less endo impurities in our pure fractions when we get them from the column. So as a result of that, we're able to mix a lot more fractions together to form a pure pool. Because of this, our recovery yield increases enormously. So from this step, we estimated that we had about 485 grams of pure product at the end of the step. And if we compare this with the recovery yield from the demonstration batch, this actually provided us with over 200 grams more product than our worst case estimation was before we launched this campaign. So we were really producing a significantly larger amount than had been estimated. So moving forward from primary purification, we then performed our ion exchange. We isolated the peptide as an acetate salt and we lyophilized. And here I've just given a summary of the results that we have from the overall campaign in the table in the middle. So we were able to obtain overall 388.4 grams of peptide from this GMP campaign. Our purification yield was now increased, so it went from 31% to 56%, and it gave us an overall yield of 47.4% versus 25.7% when we compare it to the demonstration batch. So you can see that a lot of these parameters have improved, and we have a significant amount of material for what we wanted. So we tested, of course, the peptide at the end of our campaign. So again, the identity was confirmed by MS, an amino acid analysis, and also through an elemental analysis. We were able to confirm that it was an acetate salt. In terms of residual water, TFA, and trace organic solvents, we were all within specification. We had very little left. We were able to find that there was no bacterial endotoxins, yeast, or molds, and the most important thing, of course, we were able to deliver the peptide within the client's time frame. So it was able to meet their requirements for their clinical trials. So if we compare just in terms of our worst case estimation before our GMP campaign, we'd assessed that we would probably produce somewhere between 135 to 178 grams of peptide based on our estimation. But in actual fact, we're able to produce about 388 grams of peptide altogether. So this large increase in production capacity, this really came from the gains that we're able to make in primary purification, which was carried forward into our ion exchange and lyophilization. So I hope you can really appreciate that the small changes that we're able to make upstream in terms of washing, in terms of limiting certain coupling steps, has a very, very large impact in terms of our efficiency of production over the whole GMP campaign. So that brings me to the end of my talk. Just to summarize a little bit, so we developed a GMP manufacturing process for 25 linear peptide. We established manufacturing conditions by first testing through a demonstration batch, producing about 10 grams of peptide. Through small adjustments, by just changing certain coupling times and increasing batch washes, we were able to greatly improve the quality and the purity of the peptide, the crude peptide, for GMP production. So this went from 67% to 83%. And by implementing these changes, this was able to translate into a much improved purification yield, which ultimately led us to produce over 350 grams of pure product within the specifications of the client, on time, of course. So none of this, of course, was performed by me. I was just the chemist in all of this, assisting technically. So the people that really deserve all the credit for this, of course, is our team at PolyPeptide. So I've named them here on the right-hand side. So our process development guys, our analytical validation and quality control department, our production team, which works very hard to produce so many peptides all at the same time under very, very tough conditions sometimes, and of course our project management team, our quality assurance, and our regulatory affairs team. So that's it. I'd like to thank you very much for listening, and I'd be happy to answer any questions.
Thank you very much for joining us today, Niall. Really appreciate it. First, I want to just go through for an excellent presentation. Thank you to Niall. We have received a few questions already in our inbox, and we'll give you a moment to enter your questions in the Q&A box to the left of the slides. Before we do begin the Q&A, I will run through some brief announcements. First, I'd like to thank SI Kasei Bioprocess, Wuxi, Cytiva, Phenomenex, GenScript, and of course PolyPeptide Group for sponsoring Digital Week. Now we will go back to Niall to begin the question and answer session. And Niall, I will go through.
Yes.
We have a couple questions that we've already received in here. So if you are good to go, I will go ahead and begin.
Sure.
So the first one we'll go through. For coupling completion, what was the criteria to determine completion and the coupling efficiency?
So the criteria to determine coupling completion, this is our colorimetric test. So generally perform a Kaiser test. So this is very classic in peptide synthesis. So that's for primary, after the coupling of primary amines. Or we'll also perform chloranol tests in order to assess the coupling of prolines. So this is colorimetric, so we take a small amount of resin, we perform the test, and then this is assessed under a microscope to see the color of beads. Basically, if we see the stain, if we see that certain beads are stained, then it means that the coupling is not fully complete. And if we see an absence of the stain, then we know that the coupling is within 99.5% completion. It's a colorimetric test, so we don't have absolute quantification, but it is very reliable and it works very well for us. So this is how we test the coupling completion. I hope that answers the question.
Okay, our next question goes into how do you prevent bed inhomogeneities and secure plug flows when doing percolations?
So bed homogeneities and secure. So in terms of percolation, I think I will go back to my slide because I think that would probably explain it a little bit better. So we have for percolation, of course it's animation. So in terms of our percolation, we don't have a fixed bed in performing percolation. So it's not to say, so we don't have in certain synthesis reactors, you have something at the top of your reactor, basically adjusting the bed height. So in this case, for percolation, we add our I'm going to say this again. So we deprotect our peptides in our deprotection resin solution. This is mixed and it's homogenized to ensure that everything is nice and mixed. After that, then we leave a partial volume of our deprotection solution, and then we'll add the wash solvent in a continuous flow. So in order to ensure that there is no bed immonogeneities and that the plug flow is correct, we have a pump that is adding a continuous flow of wash solvent as we go into percolation. So this is at a constant rate. So in terms of homogeneity, this is not really an issue. So in terms of the flow going in, it's fixed. And then in terms of our draining, this is also managed by a pump that's going out as well. So in terms, the whole system is basically fixed in terms of what kind of pump pressure that we have going in from either side. So in principle, then this actually controls and minimizes any homogeneities that you would have in the resin itself. So this is something, of course, that our technicians are trained very carefully in, because we need to ensure that we don't have any cracking in the resin, or that there's no that the percolation isn't performed too quickly. Otherwise, there's no point in doing it. So also how quickly the resin is, or how quickly the solution is drained, this is also very carefully performed as well, and this is judged by the expertise of the technician performing the percolation. So I hope that answers the question in terms of the percolation flow.
Next question is also about percolation. Is percolation effective for washes after AA couplings?
It is. So this is also something that has been tested by our innovation teams. This percolation technique really comes from PolyPeptide Strasbourg, and it comes from assays performed by our innovation department. So this is also something that has been tested for our after amino acid coupling. So we see that it's very, very efficient at removing residual amino acids. Generally speaking, we're not implementing it after amino acid couplings, although it is now in consideration for certain campaigns. So it's something that we would, in certain cases, consider as well. But it is effective. Yeah.
Okay, next one is the hypothesis that trace peptide and long coupling of small AAs what allows endo impurities?
So this, I would say it's not really the case, because so after each deprotection, we monitor how much trace piperidine is present then mixed in within the resin and DMF. So this is a standardized test that is actually quantified. I can't go too much into the details about how we do that, but that's to say that we really minimize the amount of piperidine that we have after our deprotection, just for this exact reason. So I don't think it's the trace piperidine that is really the root cause of the endo impurities. Actually, I think it's more related to DMF itself and its relative basicity, even though we're using a decoxy system, and some people would say that this is relatively acidic. If you go for long enough in terms of amino acid coupling, we see here, and we see it in also other campaigns, that you can have premature deprotection in very small amounts, still very small percentages. But over time, this likelihood increases, and for this reason, I think we really have the endo impurities. But in terms of trace piperidine, we absolutely quantify after each deprotection, so we have a minimal amount of this left. So I wouldn't expect trace piperidine to be the cause of these impurities.
Okay, our next question. Number one, appreciated your nice and detailed talk. Also asking about what the lead time is for the calculate scale.
The lead time for calculation of the scale. So this is performed, I would say, rather quickly. It's more, I'm not sure the exact, maybe if the question is more related to how quickly we go from the demonstration batch to the GMP campaign itself. in terms of calculating scale, I mean, once we have our final quantity, it's a simple calculation, so it's done rather quickly, and we take into consideration, of course, our production team as well, and how quickly the batch can be written up. There are certain, but I would say in terms of lead time, it's really more of a question going from demonstration batch to the GMP batch. It's really more of a question of our production schedule, and then really the priority is then the requirements of the client. So if in this case we went essentially straight from our demonstration batch to our GMP campaign within a matter of weeks, because the client's needs were such that they required the peptides by the end of the second quarter of 2022. So it was rather quick, actually. So I would say from demonstration batch to GMP batch, it was maybe 6 weeks. That's
Okay. The next question. Just trying to get the additional maybe are additional washes essential after coupling or deprotection to avoid endopeptides?
So additional washes. So in our case, we think so. We certainly think so after amino acid coupling. In this case, we initially started with well, so it depends, right? So in this case, we saw that for our demonstration batch, when we had a amino acid coupling that was complete but had to be left overnight for one reason or another. This is when we saw the formation of endo impurities. So in this case, we increased the wash procedure, and this eliminated our endo-ala impurity essentially. So I would say that it is essential in this case. In terms of after deprotection, I would say because we're implementing our percolation, we don't need to necessarily increase our washes in this case. So I would say after couplings, but maybe not so much after deprotection. But it would depend on, of course, the amino acid position.
Okay, next question is, could you please give an idea of overall project timeline from RFP to GMP batch delivery?
So in terms of this project, like I said, in terms of from RFP to GMP, so batch delivery was the end of the second quarter of 2022, and that was release delivery. So we got there in end of May, beginning of the actual activities for this campaign. In terms of development, it was sometime in October 2021. So that was the case for this campaign, so from October to May. And we had our development batch, we had analytical method development as well to take into consideration between the two, but that was also relatively quick. So I hope that gives you an idea, basically, in terms of this project, the overall timeline. But of course, this will differ depending on the amount of process development that we have to put into the project. Here, we performed a standard process development, I would say. But if you have maybe a peptide with a complex structure, or where there's very strong possibilities of forming difficult impurities, then there may need to be another iteration in between before we're going to a GMP batch, and this then will have an impact on your overall timeline. So this is something to consider, and of course this is also something that we discuss with the client before ever embarking on a GMP campaign. So if we generally try and tailor our delivery time also to the needs of the client, and try and accelerate certain activities when it's necessary for them. So this is also something that is flexible in a certain degree. Yeah.
Next one. Did you try to identify the impurities in the final product? And then how was it by synthesis or MS data?
So this is this was for a phase 1 campaign. So not all the impurities had to be identified for the need of the client, of course. as a chemist on the project, I'm always very interested to identify the impurities because it makes our job easier in the end. The more we know, and in this case, like for our demonstration batch, we were able to identify basically all the impurities in the crude, and most of them were endo impurities. So this is something that we're then able to compare our crude and our final product and see what's actually remaining at the end. So this already gives us quite a significant amount of detail. In this case, we were also fortunate that we performed a analytical development of the analytical methods between our demonstration batch and our GMP batch. So basically, our analytical development team, they worked on the demonstration batch peptide that had been purified already, and they performed a very rigorous analysis of all the impurities that you could have in the peptide, and basically tried to adjust their analytical method to basically get the best separation possible out of this. And they also studied stability and things like this of the peptide. So we have a very, for this project, we have very comprehensive knowledge of the impurities that are in the final product. And depending on the request of the client, then we will also tailor how we identify the impurities, whether they need certain impurities to be identified at the end. We can do this, and we'll also do this by mass spectrometry usually. If we're going into maybe more involved campaigns, or we're talking about maybe a phase 3 product or a peptide that's going to be really produced in large quantities, then we would consider synthesis of certain impurities to really try to pin down things that may not be necessarily 100% validated by MS or by MS-MS sequencing. It's also a possibility.
The next one, can you comment on temperature effect? Do you monitor temperature in the synthesis and washing process?
Yeah, so we monitor temperature. I'm not sure that was included in the slide, but so temperature is monitored at all stages of the synthesis, and this is something that's very important. Of course, we performed our synthesis at room temperature in both demonstration and in the GMP campaign. But indeed, this is something we're constantly monitoring because the temperature in summer or winter can also have an impact in terms of coupling efficiency and lots of other things as well. So this is something we're constantly looking at.
A few more questions we have here listed. So here you describe the development of a rather straightforward peptide. Would a similar development approach be applied to a peptide of, say, 50 amino acids with a complex structure?
Yeah, so this is that's a good question. So I would say, so this is definitely a straightforward peptide in some ways because it's linear, it's natural amino acids, and it has an N-C termini that are also standard essentially. So we would probably not apply the same development strategy for a peptide that's over 50 amino acids or that has maybe a cyclic structure, for example. So in this kind of case, then we'd really sit down and we would try and work out. Also, this would be done with this chemoinformatic analysis that I discussed earlier, and we would really try and figure out what the building sites are in terms of the synthesis of a peptide like that. We would perform probably an initial feasibility study to see just how we can make it and where really the roadblocks are. Once we've identified these aspects, we would then perform another round of synthesis where we would try and optimize for these things and make adjustments that we could then go forward and produce a demonstration batch like we did here. And only then we would perform our GMP or non-GMP campaign, depending on the needs of the client. But really, it depends on like the size and the sequence of the peptide really determine our process development and how involved it really gets. So in this case, we would probably not apply exactly the same approach as had been the case described here.
Okay, one of the final questions here. You discussed about the large excess of product obtained, of course, the peptide powder contains other substances such as water and the counter ions. How do you control for this, and how do you calculate the true content of peptide in your powder?
Yeah, so like I said, so this is described at the end of the presentation. So I just go quickly. So of course, this is also something that we discuss with the client essentially from the beginning, like in terms of their specifications. This is a very, very common thing that we test for in terms of the content of the peptide itself. So in the powder, we have water, we have TFA, we have our counter ion itself, and we have other maybe trace solvents that are used in the process. So these are all tested for either by GC or by ICP and quantified. So these are all done through quantifiable tests. And only from there, we also perform HPLC, of course, and elemental analysis. And through all these tests, we can then basically get what's called the peptide content. So this is then the actual percentage of peptide in the powder relative to all the other like the water and the counter ion itself. And from this, and based on the purity of the peptide, we then perform a calculation which gives you what's called the peptide content, which is then reported typically on the certificate of analysis at the end of the campaign. So this then gives you the true idea of how much peptide is in your final powder.
Okay, so probably will be our final question. At the beginning of your presentation, you mentioned that PolyPeptide is working on green chemistry approaches. Could you discuss that in greater detail?
Okay, so this is indeed a topic that is very important in terms of modern peptide synthesis, because of course there are new restrictions that are going to be imposed in the European Union quite soon about the use of DMF. So it's going to be, it's going to be something that's going to change, and this is something that the community needs to get together and work on, I would say. So like I mentioned at the beginning, our innovation team is working, I would say, quite hard on this particular subject. I can't go into too much detail, of course, because of for maybe some other people watching, but for instance, like we are working on aspects that's maybe trying to find other types of solvent to replace DMF, or mixtures that might be used instead of this to perform our couplings or our washing. Of course, there's more to green chemistry than just replacing solvent, but also finding ways to reduce the amount of solvent we use. So that's indeed one of the reasons why we're using percolation in our processes. But then, of course, we're also looking at ways in which that DMF can be reused or recycled in certain aspects in order to improve the efficiency of our processes and also to reduce our impact on the environment. Yep.
Sure. Yeah. Okay. So that is indeed all the time we have for questions today, all the questions that we do have for the presentation. So we do want to certainly thank you, Niall, for a great session. If anyone did submit a question that wasn't addressed, keep in mind the speaker will reach out to you directly. The session was recorded. You will receive a notification in 24 hours when the on-demand session is available for viewing. Before you log off, please do take a moment to complete the feedback form so we can continue to improve your Digital Week experience. On behalf of Informa Connect Life Sciences, I'm Mike Moriarty, and have a great day.